This video will cover an introduction to cells, including cell theory, features common to all cells, and structures of prokaryotic and eukaryotic cells. Cells were first discovered by Robert Hooke while viewing cork under a microscope, and over the next century, cell theory developed. This theory states three things. All living organisms are composed of cells. The cell is the basic unit of life, and all cells arise from pre-existing cells. There are many different types of cells with different shapes, sizes, and functions. But despite their diversity, all cells have four things in common. The plasma membrane, also known as the cell membrane, forms the outer boundary of the cell. Within the plasma membrane is a gel-like substance called the cytoplasm that surrounds the other cell contents. For all cell types, these contents include DNA for storing genetic information and a number of small structures called ribosomes responsible for making proteins. The two main cell types are called prokaryotic and eukaryotic cells. They both have plasma membranes composed of a phospholipid bilayer, but the contents they enclose are significantly different. Prokaryotes typically have a single loop of DNA and lack a true nucleus. There is no membrane separating their DNA from the other cell contents. In contrast, eukaryotes have several strands of DNA enclosed within a nucleus. The contents of the cytoplasm also differ between these cell types. Prokaryotes have ribosomes only and lack complex membrane-bound organelles. Eukaryotes, on the other hand, have both ribosomes and specialized organelles. These differences in cellular structure are considered to be so significant that they're used to separate organisms at the most fundamental level of biological classification, the domain. All living organisms belong to only three domains. Two of them, Eubacteria and Archaea, contain prokaryotic organisms. The third, called Eukarya, contains eukaryotic organisms, including protists, fungi, plants, and animals. Prokaryotic cells belong to ancient organisms, with some of the oldest known examples arising approximately 3.5 billion years ago. All prokaryotes are simple organisms, consisting of only a single cell. These unicellular organisms come in various shapes and sizes, and some, known as cyanobacteria, are even photosynthetic. For now, we'll concentrate on the structures of a typical bacterial cell. As I mentioned before, the plasma membrane surrounds cytoplasm containing ribosomes. Within the cytoplasm, you'll also find DNA. The region where DNA is located is known as the nucleoid, since there's no true nucleus with a membrane. External to the plasma membrane is a rigid structure called the cell wall. Some eukaryotic cells may also have cell walls, but their chemical composition is different than those of prokaryotes. Some bacteria may also have a protective layer called a capsule covering their cell wall, and others have long cellular extensions called flagella that function for locomotion. Eukaryotic cells are much more complex than their prokaryotic cousins, and they're typically larger as well. Eukaryotic organisms may be unicellular or multicellular, but all multicellular organisms are composed of eukaryotic cells. Some of these cells may also have flagella or other cellular extensions, but they're structurally different than those of prokaryotes. Several groups of organisms have this cell type, but we'll look at plant cells and animal cells in detail. The outermost covering of a plant cell is a cell wall made of cellulose. It provides the plant cell with structural support. Internal to the cell wall is the plasma membrane. It surrounds the cytoplasm and all its contents. Within the cytoplasm is a large nucleus that houses DNA. The nucleus consists of several different components. The round structure within the nucleus is called the nucleolus. It's responsible for producing a special type of RNA that makes up ribosomes. Surrounding the nucleus is a membrane called the nuclear envelope. It forms the outer boundary of the nucleus and is perforated by holes called nuclear pores that allow substances to enter and exit. Within the nuclear envelope is DNA organized into thread-like strands called chromatin. Just outside of the nucleus and continuous with the nuclear envelope is a system of membranes called the rough endoplasmic reticulum, also known as the rough ER. The ribosomes embedded in its surface are what give it the rough appearance. They're also responsible for its primary function, synthesizing proteins. Membranes of the smooth ER lack surface ribosomes. It's responsible for synthesizing lipids. The group of flattened sacs here that resembles a stack of pancakes is known as the Golgi body. It's responsible for packaging and transporting proteins and lipids that are produced by the cell. Mitochondria are the powerhouse of the cell, producing all the ATP that it uses for energy. The green chloroplasts are the site of photosynthesis, and the large central vacuole functions for storage of waste, nutrients, and water, holding water pressure that helps provide the plant with structural support. Notice that the vacuole takes up most of the plant cell contents, forcing the other structures out to the edge of the cell. Animal cells have many structures in common with plant cells, but there are also some key differences. For starters, you'll notice that there are several structures missing. Animal cells lack a cell wall, causing them to have a less rigid and regular appearance than plant cells. They also lack chloroplasts and can undergo photosynthesis. And though they may have small vacuoles for storage, the large central vacuole is missing. The outer covering of an animal cell is the thin plasma membrane. It encloses the cytoplasm and its contents. The nucleus is structurally similar to that of a plant cell, with a nucleolus, nuclear membrane, nuclear pores, and chromatin. You'll also find the rough ER, smooth ER, Golgi body, and mitochondria. In addition to these structures, there are a few we haven't seen before. Centrioles are responsible for producing and organizing spindle fibers, important for cellular reproduction in a process called mitosis. Plant cells also undergo mitosis, but they do it without the aid of centrioles. The lysosome is the other new structure. They can be found in some plant cells, but are much more common in animal cells. In plant cells, the large central vacuole is able to carry out their role, breaking down macromolecules and worn-out cellular organelles. So to summarize, here are the key differences between plant and animal cells. Plant cells have a cell wall, chloroplasts, and a large central vacuole. Animal cells do not. While plant cells lack the centrioles and often lysosomes that are found in animal cells. Now, let's move on to talk about another type of eukaryote, the protist. Protists are an extremely diverse group of eukaryotic organisms that have yet to be definitively classified. As such, the protists are sort of a catch-all grouping that consists of some organisms more closely related to plants and others more closely related to fungi and animals. Most unicellular eukaryotes are included in this group, but others, such as the kelp on the right, are multicellular. The representative protist that we'll be studying is Physarum polycephalum, commonly called slime mold. It grows in a highly branched network that consists of a single huge continuous cell. This cell is multinucleate, meaning that it contains many nuclei, but they are too small to be visible with a compound microscope. Its unique cellular structure makes it an excellent specimen for viewing the process of cytoplasmic streaming. Cytoplasmic streaming is the flow of the liquid component of the cytoplasm, called cytosol, that can be observed within large eukaryotic cells. This flow aids in the delivery of substances throughout the cell. As part of your online lab activities, you'll view a demonstration of this process and use it to complete the cytoplasmic streaming demo exercise in your lab manual. Now it's time to try it on your own. Use the plant cell diagram on page 69 and the animal cell diagram on page 71 to label cell parts on the model photographs provided on eCampus. Be sure to familiarize yourself with these models, as they're the ones you'll see on your lab exam. After you're comfortable with the models, use your lab manual, textbook, and online resources to complete Table 5.2 on page 73 of your manual. Finally, you'll use the cytoplasmic streaming demo on eCampus to complete exercise 5.2a. The learning objectives you're responsible for are listed in your lab manual at the beginning of Laboratory 5. The ones specific to this video are listed here. Make sure you understand all terms listed in bold and italics, and be able to describe similarities and differences between the different cell types. You should also be able to identify and describe the function of the major cell components. Finally, you'll observe cytoplasmic streaming in a representative protist cell. These objectives will be tested in your upcoming quiz and lab exam. Contact your lab instructor if you need any help or anything's unclear. Good luck!